Welcome to this podcast from Neurogastroenterology and Motility. It publishes original research and topical reviews on basic and clinical aspects of gastrointestinal sensation and motility, as well as brain-gut interactions.
So welcome everybody to this month's podcast from Neurogastroenterology and Motility. My name's Adam Farm and I'm a gastroenterologist at the Wingate Institute in London. This month it's my real pleasure to welcome Professor Pieter Vanden. Pieter studied bioengineering and obtained his PhD in 2000 at the University of Leuven and currently works at the Laboratory for Enteric Neuroscience and Cell Imaging Core at the Translational Research Centre for GI Disorders at the Catholic University in Leuven. So Pieter, many thanks for joining us on the podcast this month, and congratulations to you and your co-authors on your paper entitled Fructose Consumption Impairs Serotonergic Signalling in the Murine Enteric Nervous System. So if I could start by asking you what has previously been known about diet and neuronal survival and function.
Okay, thank you very much for having me. I think what is interesting to realize is that whatever we ingest, take in, can actually influence our enteric neurons, or the neurons in the intestine. So previously, people have done studies where they used high-fat diets, where we see that some neurons actually disappear after very high content, high-fat diets and high caloric intake. So also a study by Paul Bertrand in 2011 showed that after Western diets, high-fat diets, they have increased TPH1 expression, more enterochromaffin cells, and decreased serotonin transporters. So there's something known already. Our idea was to really do a study about just only one molecule and see whether that was actually able to influence enteric nervous system and enteric nervous system function.
So why has fructose intake changed over the recent past?
Yeah, actually in the last century the total fructose intake has increased from 20 to 100 grams a day, so about 20% of the daily energy intake. And especially with the invention or the advent with whereby high fructose corn syrup was made, or the technology that was invented to make that high fructose corn syrup. This was used as a sweetener, and it's fairly easy, fairly cheap to produce this from cornstarch. And therefore, in the late, I think the technology was probably invented in the late 50s, 1950s, and from the 1970s, 70s onwards, this fructose high fructose corn syrup was used to sweeten industrial foods. So especially in soft drinks and in many industrial foods, this is just added for caloric content, sweetening the beverages and foods. So that's why we all eat way more fructose than 100 years ago.
That's very interesting. I think it's probably reasonably ubiquitous across Western diets now. So what was the objective of your study?
Well, as I told earlier, for us it was we wanted to know whether one single component of these diets in the regular Western diet animal studies, high-fat diet animal studies, people feed mice these high-fat diets with a lot of starch and a lot of fat, lard, etc. We were interested to know whether specifically the fructose molecule that is now in all these soft drinks, whether that one on its own was able to induce these changes already. Problem with these high-fat diets is that the animals have a lot of other changes as well. Obviously metabolic changes, they gain a lot of weight. Their fur is incredibly greasy, so they actually do not succeed in keeping themselves clean. So also in the interaction within the mouse colony, it's completely different. In our study, actually, these mice, we feed them 6 weeks of fructose, or 4 weeks of fructose in one case, and we see hardly any changes in terms of caloric intake. There is no change between the control colony and the subject colony, where energy intake is the same, blood glucose stays the same, body weight evolves in the same way. So it's a far more subtle dietary intervention, yet we do see effects on the neuronal activity already.
So what methods did you use in your study to address your objectives?
So obviously to control what the mice were behavioral things. So obviously we controlled for total energy intake, we measured blood glucose, body weight, and then we used a number of techniques to see whether there was an increase in permeability, because there have been a couple of studies where mice were exposed, or rats I think it was, were exposed to 8 weeks of high fructose, and they developed steatosis and a lot of permeability changes in the intestine. So we wanted to check that first, and we found that there were zonocludins 1 and occludin mRNA was reduced in the population that was exposed to fructose. However, we were not able to measure any significant changes in permeability. So we believe that maybe this could evolve, but actually our measurements that we done then afterwards were before any major involvement of permeability changes. And most importantly, we used imaging. I with my team have developed a number of fluorescent imaging techniques to monitor activity in the enteric plexus and the submucous plexus in this case. So we peel the intestine of these mice of the two populations, and we expose the neurons to different stimuli, and therefore using calcium imaging we can actually assess their activity, whether they respond differently more or less to the known agonists.
That's fascinating. So what were your key results to emanate from your study?
Well, I think the most important thing is that, as I said, before we see any dramatic metabolic or permeability changes, that this little molecule on its own is actually able to change already neuronal function. And that was also derived from some other studies that we've seen from the central nervous system that suggest that high fructose intake would actually have cognitive effects and reduce the neuronal activity in the central nervous system. Was seen from bad performances in water maze tests of mice fed on fructose. So the idea was to see whether indeed in the enteric nervous system we had something similar. And when we depolarize the neurons, which is just a general tool to see how functional the neurons are, we have already reduced responses to blunt depolarization. And when we check them with serotonin being the key molecule that translates intestinal or luminal signals to the enteric nervous system, we see that also there we have less responses or less numbers of neurons respond, although the submucous plexus looks absolutely normal macroscopically. And also the transients that were induced, the calcium transients that were induced by serotonin were a lot lower than in controls. And interestingly, if we feed the mice 2 weeks of water after the 4 weeks of fructose, we readily reverse this effect. So it's really a dietary intervention that is subtle, but it shows already that very quickly we can modulate the activity of these neurons in the intestinal wall.
That's very interesting. So how do you explain the reduced serotonin sensitivity that you saw in the study?
Well, that's actually not completely clear. It can be explained in two ways. Or there is, of course, less serotonin, and therefore the neurons are less capable of responding to whatever serotonin would be available. But a little bit more likely is the fact that there is actually an or an excess of fructose during these weeks that they have been fed fructose. We see from the reduced amounts of serotonin transport that this could be a possibility. So it's possible that there is just more serotonin released from the enterochromaffin cells, less taken up because of fewer serotonin reuptake transporters, and therefore the neurons might be baited in serotonin for too long. And maybe that is the reason why, if we then apply exogenous serotonin, we have fewer responses. So that is something that could be further explored in the future what the molecular nature is of these reduced responses.
Absolutely. So what would you regard as the limitations of your study?
Well, this is actually one of the limitations personally find, is that we were not able to find the molecular pathway or the really cellular sequential event. So it's obvious that there is an increase in GLUT5, which is in the intestine, an increase in GLUT5, the transporter of fructose. So fructose definitely does something to the intestinal wall. Unfortunately, we weren't able to pinpoint where exactly that was due to the lack of good antibodies to identify where exactly spatially that happened. So that's a bit an unfortunate thing that we haven't been able to do that. And the other thing is that it's a study where after the intervention we have to sacrifice the animals, so it takes quite a number of animals, and it would have been easier, nicer, better if we would have been able to monitor that in a live way while the animal stays alive. At this moment, not possible with our techniques, but that is of course a bit of a limitation, that you need larger populations to see an overall effect, and it's very hard to post hoc then try to find what the molecular pathways was. Not all the tools are available to actually identify that, especially in the mucosa, where imaging is quite challenging because of autofluorescence, etc., etc. I think we have to be clever in the future to invent new ways to work on that particular point.
Absolutely. So I appreciate this is a very exploratory question, but what do you feel are the potential translational implications for humans, and particularly those eating Western diets?
Well, it's for me, it's very obvious that these Western diets with high fat and are definitely doing much more to your body than only affecting your enteric nervous system. But if you once focus now on this nerve function, it's clear that even for a very short period, that is in our case 4 to 6 weeks of fructose intake, which would resemble drinking a couple of soda beverages every day, a couple of Cokes and these sweetened beverages every day, that this already affects neuronal function. So I think if knowing that, seeing how reversible it is, maybe we can use this kind of knowledge for people without severe diseases, but having sometimes some annoying pain, itch, or whatever thing that bugs them in a relatively mild, but just by changing the diet during these weeks for a while with particular additives, or that this would help relieve their problems, and not so much solve a disease, but that we can easily intervene if we know a little bit more about these sugar or other containing foods. And obviously it's clear that that is best for cellular health to use a varied diet, and then I think you keep your neurons and the whole mucosa neuronal interaction as flexible as is. Otherwise you just lose the flexibility for it to react to other substances that you might ingest.
Absolutely. I think as you rightly say, the translational implications are difficult based on your animal data. But where do you think the knowledge gaps lie currently in the field, and how do we go about to resolving these?
Well, I think it's important to, first of all, if we want to relate this or translate it to humans as well, is that of course our study was done in mice, and mice obviously don't have the same way of feeding. They're normally not exposed to fructose. We can choose to eat or drink fructose whenever we want. So a human study using maybe biopsies, if that is ethically defendable, then that might be giving us a hint whether the same principles apply there. I don't think it's that difficult to have a couple of volunteers on a fructose diet or a higher fructose diet and see whether As you probably know, we also can do these imaging in submucous plexus in biopsies from the duodenum or small intestine. We can measure activity of human neurons there. So it would be good to test whether that is indeed the case also in humans fed with high fructose beverages or food. That is one thing. And on the other hand, in terms of mouse work, I think it's very interesting to try and find out whether it is a specific cellular metabolic effect, whether the cells actually get a bit lazy in that they have a lot of energy in excess, so they don't need to be really sharp to maintain normal cellular function, so they get a bit lazy, or whether it is instead of just one single cell, or whether it is a circuit. So I think we have to be clever in designing methodology to find whether the specific nutrients, fructose in this case, whether they activate a specific pattern, a specific circuitry within the intestine that then as a whole might be influenced by dietary interventions.
So with that, I'd like to thank you and your co-authors for a really excellent paper and also assisting in this month's podcast, and I'd like to thank our listeners for tuning in. I look forward to welcoming you next month for another instalment.
Further information about this paper can be found on the journal website.